Hi, this is Rethik, your biology subject matter expert from the MemoNeet. And once again, welcome to the biology series. So in this video lecture, we'll be concentrating on the transforming principle. That is how the genetic material has been, getting transferred from one to the next generation. Based on these all things, we will be studying some major experiment which was conducted in order to prove how this transformation principle is going and how the genetic material is flowing from one generation to the another generation, right? So without delaying on the topic, we'll just move on to the major one. So what happens is after the discovery of the new, what do you say that the nucleus and the genetic material, but the thing was not everyone were able to understand how this genetic material has been transferring from one generation to the other generation. So for that, in 1928, the Griffith he conducted a series of experiment with this specimen that is Streptococcus pneumoniae. So what is this? It is a kind of a bacteria which is actually responsible for the pneumonia. Right. So what happened is when the bacteria were actually grown on two different plates, okay? So this pneumonia bacteria were being grown on two different plates here or Petri plate. So and it was cultured. So some produced a smooth, somewhat shiny, kind of thing. So shiny colonies were produced like this. So smooth, very shiny colonies were produced, and some, they produce somewhat hard colonies, rough colonies. Okay. So the one which actually produces smooth, shiny colonies, so they named it as yes strain, and with other which actually produce the rough colonies, they named it as R strain. Okay. So yes strain and R strain colonies. So this because this S strain, the colonies or the S strain bacteria, they had a kind of coat or they were being covered that is called as mucus or polysaccharide. Okay, so that means the colony were being coated by the polysaccharides. This is one point. Whereas in R strain, they did not absorb such coat. Okay, so this is the difference between S strain and R strain. Yes strain is a smooth strain, and it is what? Smooth, shiny colonies, where R strain is rough colonies. And yes strain, it had this mucus, that is polysaccharide coat was there in yes strain, whereas in R strain, you do not find such coat. So then what he did, he conducted a series of experiment where he took the yes strain bacteria first. Okay, so these are the bacteria colonies that you can observe here. Yes, so these are the bacterial colonies that you can observe here. So one is having what? The rough strain, and the another one was the smooth strain. So first what he does is he is going to take up this R strain, that is the rough strain bacteria. Then what he did basically, he had, sorry, yeah. So we had one is yes strain was been injected to a mice. Okay. And another one is R strain was injected to again mice. So now you can see that R strain was injected to mice and yes strain was also injected to mice. What was the result now? When a yes strain, that is the smooth strain, when it was injected to mice, right? So as a result, the mice, they were died. So mice died. That means when a yes strain was actually injected to the mice, so the mice was dead. Whereas when the rough strain, R strain was injected, so the mice did not die, so it continued its life. So that means resulted the mouse was still healthy when R strain was actually injected to the mice or mouse. So one thing was the Griffith was able to kill the bacteria, and the next what he does is he conducted that where he killed the bacteria. Okay, both the bacteria was been killed. So he conducted that R strain bacteria at somehow. So after that, what he does, he killed the bacteria. Then he observed that heat killed a yes strain bacteria. Okay, so you are seeing here. So heat killed yes strain bacteria was now again injected to the mouse, healthy mouse now. Okay. As a result, what he'll be observing? What was the result he saw? So again in yes strain, which was heat killed. So it was heat killed, right? So when it was injected to the mice or mouse, so mice lived. And again, R strain also, it was very normal. The mice, well, anyhow, it is going to leave. So that what he did, what he conducted is he took the S strain bacteria, bacterial colonies, he heat killed it, and later on that killed bacteria. So that S strain, heat killed smooth virulent, bacteria. S strain was again injected to what? An healthy mouse. As a result, mouse lived. That means mouse was again healthier. So later on, what he did is he took this heat killed, okay, S strain and R strain, which was non-virulent bacteria, right? R strain, that is pneumonia, Streptococcus pneumoniae, and he have mixed it like in a centrifugation. Okay, as a result, and obviously both were being brought together. Then he injected a mixture of this heat killed S strain and lived or live R strain. Okay. Then when he absorbed it in the mice died. So now the question comes is it is live. It is in what condition now? Live condition. And it is dead condition. That is heat killed. Okay. So this is live condition. We just got to know that before when this R strain was been injected, the mice did not got any effect from that thing. And that means R strain is okay. Okay. That is not a virulent bacteria. Like why it doesn't produce any disease. That is what we got to know. Now this heat killed strain, by seeing that experiment when yes, the strain was heat killed and it was injected, again we got to know that okay, now the mice was able to live even though when it has been injected by yes strain. That means heat killed bacteria will also not affect the mice. So that is what we have understood. But here in the next experiment, when he mixed that heat killing, yes strain bacteria with the rough yes strain, which is live in condition, as a result, what happened? The mouse died. Okay, as a result, what happened? The mouse died. So by this thing, one thing he got to know that, okay, he took the blood sample also and he found that yes strain in blood sample from the dead mouse. So now it is something confusing for him, and later on he concluded that this R strain bacteria had somehow been transformed by the heat killing yes strain bacteria. That means R strain bacteria, which is in a live condition, right? So they got transformed into yes strain bacteria, which was actually what? Been heat killed bacteria. So that means he got to know that there's somehow this transforming principle that is might have been applied in this case. Okay, that is what he got. He came with the conclusion that transformed from the heat killed yes strain, that is this rough R strain got transformed into yes strain. That is what he concluded. Okay, then there must be due to the transform. So what might be the reason? Again, he gave a conclusion saying that because there might be some genetic material which has got transferred to the R strain by the S strain, and S strain, since it is dominant, so what happens? Obviously, it is going to express itself in the next generation. Say like we have, so say like we have two bacteria here. So one is S strain bacteria and R strain bacteria. Okay. Say like this is R strain and this is S strain. Just to denote, I'll write the genetic material in red. So now this S strain is heat killed for sure. But when they were mixed, right, as a result, this genetic material might have got transformed. This genetic material might have got transferred here. And since it is dominant, right, yes, strain genetic material is dominant, it might have expressed itself in this, bacteria. And obviously, the live bacteria will replicate to produce more number of bacteria. So in any generation, so obviously that the genetic material of yes strain might have expressed. That is what the conclusion they come up with. Clear with it? So this was an experiment by which they got to confirm that the genetic material is what it is expressing. So there is a characterization of the transforming principle. Prion, with in 1933 to 44, these are the scientists who worked on this specific experiment to know how this transforming principle is knowing and their characterization. So one thing we know that the genetic material was thought to be a protein till that day. Okay. So they were to determine the biochemical nature. That is, whenever we are determining any proteins, we need to understand their biochemical nature. So what is the composition of them? Then only we will be understanding what the transformation principle. So now the Griffith, he proposed a transforming principle. So they started to work to determine the biochemical nature of the transforming principle, which was been, that principle which was given by the Griffith. Right. So one thing what they did is they purified this biochemical such as protein, DNA, RNA, etc. from the yes strain bacteria or yes strain heat killed yes cell to see which one could transform. So like what will transfer either the protein, DNA or RNA, which is going to transfer to the from yes strain, heat killed yes strain to R strain, that is our living bacteria. Okay. So they discovered that DNA alone from yes strain bacteria causes the R strain bacteria to become what? Transformed. So they concluded that the DNA alone is also sufficient, okay, to transform from yes strain bacteria to R strain bacteria. And it is enough to, to make this change, transform the R strain bacteria to X strain bacteria. This is clear. So then they also discovered that the protein digesting enzymes, that is protease, and uh, RNA digesting enzymes, these two enzymes did not affect the transformation. That means inside after transformation, that protein digesting enzymes and RNA digesting enzymes were not affected in the bacterial cell. So the transforming substance was not a protein or RNA. That was clear to them. Okay. So digestion with DNAs did inhibit the transformation, suggesting that the DNA causes the transformation. Only which was affected, the digestion with DNAs or DNAs. Okay. That thing was being inhibited. So that was the one which was inhibiting the transformation. So which suggested that not the protein, because there are nowhere this protein digestion enzymes or RNA digestion enzymes, digestive enzymes were being affected. So only DNA digestive enzymes was affected. Or it was inhibiting the transformation. So they got a clarity that DNA causes the transformation. So they concluded that the DNA is the hereditary material or the genetic material, but not all biologists were convinced with this theory. Okay. So, but still, later on, many controversies case whether it was always the DNAs or DNAs. Okay, which what might be the differences and whether RNA could also be a genetic material and all. So we will be learning about it one by one. So if I conclude this as the DNA, which is actually a genetic material, okay. So if the DNA is to be a genetic material, so a unique proof experiment was conducted, okay, that the DNA is a genetic material. So which was an experiment which was done by these two scientists in 1952. So they work with the viruses that infects the bacteria by using the bacteria, infected bacteria, the virus which normally affects the bacteria, which uses bacteria as host, they are actually called as bacteriophages, right? So that experiment was done. So the bacteria here, what happens? You will be seeing here that uh, the bacteriophage that you're seeing, right, which normally goes and attaches. So we can actually see here that the bacteriophage, which normally comes and it is going to attach to the bacteria, and its genetic material, as you can see here, that its genetic material gets enters into the bacterial cell, right? Can you see here there is a genetic material? Now this genetic material is getting entered into the bacterial cell, right? And the bacterial cells treats the viral genetic material as its own genetic material. Okay, so starts to manufacture more viral particles. It starts to manufacture more viral particles or virus particles. So therefore, this Hershey and Chase, they worked on this experiment, okay, to know how this entry and in what way the genetic material has been determined and all. So in the experiment, the only thing they did was they actually grew some of the viruses on a media that contain what? A radioactive phosphorus and some in the media, and another media containing radioactive sulfur. Okay, so one was, they took two media. So in one of the media, what they did is they have grown the viruses. Okay, they contain what? A radioactive, what you can say, phosphorus, and in one of the media contains radioactive sulfur. So virus which actually grow in the presence of this radioactive phosphate, right? So they contain radioactive DNA. So they had what? The radioactive DNA, but not radioactive protein. So they had what? Radioactive DNA, but not protein. Why? Why not protein? Because DNA contains what here? Phosphorus, but protein doesn't contain the phosphorus here. So similarly, the virus which was actually grown on this radioactive sulfur, the sulfur, so they contained a radioactive protein here. So they contained what? A protein here, but not the radioactive DNA. So they did not contain DNA because DNA does not contain sulfur. So obviously DNA cannot grow on sulfur media. So where proteins cannot grow on phosphorus media. So this was clear now. Now, this radioactive phage, that is the bacteria phage, they were allowed to attach. Both the bacteria were allowed to attach to one E. coli. Okay, they were allowed to attach to the E. coli bacteria. So, whereas the infected and they made to leave it as an infection process. So, the bacteriophage, they came, they infected. Then what happened? The viral coats were removed from the bacteria. Then the viral particles were separated from the bacteria. So that means the bacteria was infected and all we got to know the infections, everything was done. So now the bacteria were being separated and the viral coats, the virus which were attached to the bacteria, they were removed and later they were separated. The viral particles were separated from the bacteria by spinning them in the centrifugation. So after that, what happened? The bacteria which was infected with virus, okay, that had the radioactive DNA were radioactive, obviously. So that had a radioactive DNA were radioactive, indicating that the DNA was the material passed from the virus to the bacteria. That means here the DNA is a genetic material which was passed from the virus to the bacteria. Whereas the bacteria that were infected by the virus, which had what? Radioactive protein, were not radioactive. So when they have subjected it, they got to know that the DNA, so the bacteria which had a DNA, because the viral, it passed its DNA to the bacteria. So it was showing the radioactive. That is, the radioactive DNA showed radioactive thing. So that means it has DNA. And the bacteria which was where this protein, was being subjected to the next to the bacteria by the virus, so it did not show that radioactive thing. So that means it is protein. Now this indicates that the protein did not enter the bacteria from the virus. Okay, so DNA is therefore the genetic material that is passed from the virus to the bacteria. That is what they have concluded. In summarizing it, what I can say you is there is a virus which is having two genetic material. So one could, you can name it as the DNA here. Yes. So one could be the DNA, could be a genetic material. Sorry, what happened? Yeah, one could be the DNA, could be a genetic material, and the another, the protein could be the genetic material. So they led it to, infect to the bacteria. Once infection was done and the genetic material was passed from the virus to the bacteria, so they have separated the viral capsule, viral materials from the bacteria through what? Centrifugation technique from both. And as a result, after centrifugation, they led those bacteria to, um, obviously differentiate or to replicate and all. And they saw that the phosphorus detected in the cell. So in the cell, phosphorus were detected, and in the another cell where the the virus which had the radioactive DNA, right? So that was passed up to the bacteria. That is what they have detected. But the virus which has the sulfur or radioactive sulfur, so that was not, that is, the protein was not passed to the bacteria. That is, if it did not show that radioactive sulfur detection was not seen in the cell, that means obviously the protein was not passed to the bacteria. So therefore, finally, they have concluded that if the radioactive phosphorus has been detected and not the sulfur is detected, because protein consists of sulfur, DNA consists of phosphorus. So phosphorus is detected, that means the bacteria is having DNA. So that means the DNA has been transferred from the viral to the bacteria. So whereas this sulfur, radioactive sulfur, was not detected, which shows that the protein from the virus was not transferred to the bacteria. So therefore, which is showing the transformation here, transforming thing here, that was bacteria. So finally, they have concluded that the DNA is actually the genetic material which has been, transferring from one generation to the another generation. This is how they concluded, whether it could be the DNA or RNA. Okay, so this was all about the basic principles of the search for genetic material, basic thing, an experiment, which is, so the questions will always be in a twisting form. So they will always ask you in a twisted form. So make sure the concept, like whenever you're going through this specific topic and this specific chapter, concept has to be clear. You should know which bacteria is having what. Okay, so in both the experiment, where in actually in all both the experiment, that is in the transforming principle also, and when you are going with this genetic material as a DNA also, you need to understand and have this specific thing in mind. What experiment defines what? Sometimes, even in theory, if they ask you to write some genetic material experiment related, so most of the students get confused with two things. One is what do you say, that R strain and S strain experiment, and another one is hershey-chase experiment. So they get confused and they'll think, they'll interchange and they'll write a different answer and they'll come. So make sure that you are clear with the concepts. So this is all about the video. So yes, if you have any doubts regarding the biology, you can, contact me out to the comment section. This is one point. And the second thing is, you can also, contact me through the Telegram channel, which is already in the description box. And very important thing is, if you're planning to purchase Elite Plan or Brahmastra test series, yes, you can go for it, as you are getting it in a very low cost and a very reasonable cost. So make use of it and keep practicing more number of MCQs. Once again, thank you for watching the video, and don't forget to subscribe, like, comment, and also share with your friends. Thank you.